Hello everyone, this is Shitiza from Department of Biochemistry, Indian Institute of Science. I'll be talking to you about an extremely important and very interesting form of cell division called meiosis. You would recall from your school biology textbooks that cell division is a critical process in growth, development, repair and reproduction. You have been taught at great length about another form of cell division called mitosis in Professor Shikha's previous lectures. So I would start by asking you, why do you think an entirely different form of cell division called meiosis occurs while mitosis is already there? Now imagine, when you were a zygote, you received half the number of chromosomes from the egg cell which came from your mother and half the number of chromosomes from the sperm cell which came from your father. Now hypothetically, if the egg cell had diploid number of chromosomes, that is 2n, and the sperm cell also contained diploid number of chromosomes, that is 2n, the fusion of the egg cell and the sperm cell would form a tetraploid zygote, which would be the abnormal number of chromosomes and is not found in human baby. Therefore, an entirely different form of cell division called meiosis evolved, which made sure that the number of chromosomes in the egg cell and the number of chromosomes in the sperm cell is half the number of chromosomes present in the zygote, which is diploid, that is 2n, such that the ploidy is maintained in the offspring. Now, as you can see here, the difference between mitosis and meiosis occurs in three important aspects. Starting from a diploid cell which has a ploidy of 2n, in both the cases, DNA duplication is a common step, and after DNA replication, the number of chromosomes stays the same in both mitosis and meiosis. However, the content of the DNA is now doubled. I've shown here 4 chromosomes per cell, 4 chromosomes shown in different colors. When the DNA is duplicated, you see that the chromosomes are shown in form of 2 chromatids, which are called sister chromatids, which are joined together at a point called centromere. When mitosis and meiosis are compared, after DNA replication, in the mitosis process, there is only one round of chromosome segregation. However, in meiosis, one round of DNA replication is followed by 2 rounds of chromosome segregation, which results in formation of 4 daughter cells, as you see here, with a ploidy which has half the number of chromosomes as compared to the parent cell, which had 2n number of chromosomes. This is in contrast to mitosis, where only 2 daughter cells are formed, which have a ploidy which is exactly the same as the parent cell. The second very important aspect is, during the meiosis process, the 2 homologous chromosomes, that is the version of chromosome one received from the mother and one received from the father, pair together, and during the first meiotic division, which is called meiosis 1, these homologous chromosomes segregate, and during the second meiosis division, which is called meiosis 2, the sister chromatids segregate. I've shown here 2 pairs of homologous chromosomes. In purple and in green is one pair of homologous chromosome. In black and in pink is another pair of homologous chromosome. After meiosis 1, one of the daughter cells contains one pair of homologous chromosomes which has been segregated, and another daughter cell contains another pair of homologous chromosome which has been segregated, and this is called a reductional division. Meiosis 2 is almost similar to mitosis in a way that the ploidy does not change from these 2 daughter cells to the 4 daughter cells that are formed here. The third very important aspect of meiosis, which is different from mitosis, is when the homologous chromosomes pair, there is an exchange of genetic material between the 2 homologous chromosomes. This process is called crossing over, and it occurs via DNA recombination process. Let's dive deeper into the different stages of meiosis. Just like in mitosis, there are different stages of the meiotic process, starting from a diploid cell in the G1 phase, as you see here. In S phase, the DNA is duplicated, which is followed by a G2 phase where the homologous chromosomes start to align with each other. After G2 phase, meiosis 1, which is also called a reductional division, starts. Prophase 1 of this meiosis is different from mitosis in a way that in meiosis the prophase 1 is quite long because the homologous chromosomes need to pair together and DNA recombination has to occur. In prophase 1, the homologous chromosomes pair and crossing over happens. It is followed by metaphase 1 where the paired homologous chromosomes align at the equator, which is followed by anaphase 1 where the homologous chromosomes segregate and each of the homolog is segregated to different poles, opposite poles. Anaphase 1 is followed by telophase 1 and cytokinesis 1 where the cytoplasm divides. Now note that since the green chromosome and the purple chromosomes are homologs of each other, they will never be segregated towards the same pole. Similarly, for the black chromosome and the pink chromosome, they won't be segregated towards the same pole. However, it is entirely possible that the green chromosome here is segregated with the pink chromosome here, and which will lead to the purple chromosome here segregating with the black chromosome here. Therefore, this is one of the combinations that I have shown here. The other combination is equally possible. This is one level where merely on the basis of chromosome segregation, different types of gametes can be formed and genetic variation is maintained. Now, since I have shown here only two pairs of homologs, you should remember that in human cells there are 23 pairs of homologous chromosomes, which would lead to an enormous number of possible combinations and therefore an enormous amount of genetic diversity merely on the basis of chromosome segregation. Now, meiosis 1 is followed by meiosis 2. Meiosis 2 is also called equational division because it is very similar to mitosis. Now that the chromosome number in the daughter cells has already been halved during meiosis 1, during meiosis 2, 4 daughter cells are formed from these 2 daughter cells and the chromosome number now stays the same. During prophase 2 of meiosis 2, new spindle forms around chromosomes as you see here, and in metaphase 2, these chromosomes align at the equator. As we saw in anaphase 1 of meiosis 1, homologous chromosomes segregate towards opposite poles. During meiosis 2 and anaphase 2 in meiosis 2, sister chromatids segregate towards opposite poles. So the connection between the sister chromatids that was characterized by the presence of centromere here, this connection is lost and the sister chromatids are pulled towards opposite poles, as you can see here. Now, as we saw in meiosis 1, there were different combinations possible for the segregation of homologous chromosomes, and that led to formation of different types of gametes. Similarly, in meiosis 2, when the telophase and cytokinesis 2 occur, the cytoplasm divides and 4 haploid cells are formed. Different combinations of segregation is possible, and this will lead to different combinations formed in these haploid cells. This is another level of genetic variation, and this is one very important reason why meiosis evolved in the first place. The meiotic prophase is a beautiful process in itself, and it's so intricate that it has been subdivided into 5 different stages, as you see here. The first stage is the leptotene stage. Now, after the DNA has been duplicated in the S phase, the DNA still exists in form of thread-like structures called chromatin. During leptotene stage, the chromatin condensation occurs and now chromosomes are formed. I've shown here one pair of homologous chromosomes where the first is purple in color and the second is green in color. As you see here, chromatids from one of these homologs are called sister chromatids, while chromatids from two different homologs in the pair are called non-sister chromatids. The leptotin stage is followed by zygotine stage. In the zygotine stage, the nuclear membrane starts disintegrating. Also note that the homologous chromosomes have started to align with each other. Now this alignment of the homologous chromosomes leads to formation of a structure which is called a bivalent. The pairing of the homologous chromosomes or the coming together of these homologs is facilitated by formation of a proteinaceous complex which is called a synaptonymal complex. The synaptonymal complex is proteinaceous in nature and is composed of proteins which are responsible for not only keeping these homologs together but also regulating the DNA recombination between these homologs. The zygotine stage, where the formation of the synaptonymal complex is still immature, is followed by Packettine stage, where the synaptonymal complex formation is matured. These homologs are glued together such that the distance between them is now almost 100 nanometers. This is an ideal situation for DNA recombination. Note that DNA recombination should only occur between non-sister chromatids and not between sister chromatids. This process is also highly regulated and it involves many proteins of the synaptonymal complex itself. The points of exchange of genetic material between the homologs is marked by these points of association and these are called chiasma. Now I have shown one homolog pair here and one point of crossing over, one chiasma. However, there can be multiple points of crossovers between one pair of homologs. However, the number of crossovers between one pair of homologs is highly regulated. So the Packettine stage is marked by the formation of the synaptonymal complex and crossing over between homologs. Packettine stage is followed by diplotin stage. Now after that exchange of genetic material is over between the two homologs, it is essential that the homologs need to be separated from each other and this glue needs to be removed. In order for this association between the homolog to be removed, there is disassembly of synaptonymal complex. So the proteins that were initially recruited between these homologs are now de-recruited from these homologs in order for the homologs to separate during anaphase 1. During diplotin stage, the crossing over is completed and the synaptonymal complex disassembly begins. However, the homologs stay connected only at the points of crossing over, which are the chiasma. The separation is completed during the dikinesis stage, where the homologs completely separate from each other and the nuclear membrane is completely dissociated. Note that we started with a parental combination of genetic material here in purple and green, and we ended up with a slightly or majorly different combination of genetic material in the daughter cell here. And this is an extremely stunning process. We'll see how DNA recombination occurs, but before that, we would see how the synaptonymal complex formation happens. What you see here is a schematic of the synaptonymal complex as observed in budding yeast by scientists during meiosis. We have already discussed that in order for the two homologous chromosomes to stay together, a proteinaceous structure called the synaptonymal complex assembles between them and keeps them in a close proximity such that the distance between them is almost 100 nanometers. Now let's see what are the protein components that make up the synaptonymal complex in budding yeast. What you see here are chromatin loops from one homolog in the pair, and these are the chromatin loops from another homolog in the pair. The first type of elements are called lateral elements. The lateral elements are majorly composed of two proteins called HOP1 and RED1. By research, it has been shown that HOP1 protein can actively bring together two different DNA molecules and keep them in close proximity, while RED1 is also essential to maintain this glue, and both of these proteins are absolutely essential in regulating recombination in such a way that they bias the DNA recombination between interhomolog rather than between intersister chromatids. The second type of element, called the central element, is composed of two different proteins called ECM1 and GMC2, and the final structure is kept together by a third type of filament called transverse filament, which is composed of a protein called ZIP1 in budding yeast. Now it's very important to note that the entire assembly of the synaptonymal complex is evolutionarily conserved. By that I mean that the proteins that are involved in the formation of the synaptonymal complex have homologs from yeast to mammals. So a synaptonymal complex is not only evolutionarily conserved structure, it is also functionally conserved. We have also seen that during anaphase 1, after the DNA recombination is complete, the homologs need to segregate from each other. Therefore, the timely loading of these proteins to the chromosome axis is essential for assembly of the synaptonymal complex, and the timely removal of these proteins from the chromosome axis is absolutely essential for the disintegration of the synaptonymal complex in order for the homologs to separate during anaphase 1. Now that we understand the structure of the synaptonymal complex, let's look at the dynamics of the synaptonymal complex in budding yeast in context with the different stages of meiotic prophase 1. What is shown here in white are two sister chromatids from one homolog in the pair, and what is shown here in blue are two sister chromatids from another homolog in the pair. Chromatids from two different homologs in the pair are called non-sister chromatids, and recombination is biased towards recombination between interhomolog rather than intersister. During the zygote stage, we know that the synaptonymal complex assembly starts by loading of the proteins that we just saw. During Packettine stage, the synaptonymal complex assembly is matured and the two homologs are brought close together and crossing over occurs. Now, in order for the homologous chromosomes to segregate during anaphase 1, synaptonymal complex disassembly is equally essential, and this is brought about by removal of the proteins that were involved in maintaining the synaptonymal complex here. The synaptonymal complex disassembly is brought about during diplotin and dikinesis stage. In case the synaptonymal complex disassembly does not occur properly, it can lead to a situation where one of the daughter cells would get an abnormal number of chromosomes, or one of the daughter cells would not get any chromosome at all, and such situations are called situations of non-disjunction. Approximately 10 to 30% of fertilized human eggs contain an abnormal set of chromosomes, and this is a result of non-disjunction of chromosomes during meiosis. Non-disjunction or missegregation of chromosomes can occur either during meiosis 1 or during meiosis 2. You would recall that during meiosis 1, homologs are the ones that segregate, while in meiosis 2, sister chromatids are the ones that segregate to the opposite poles. Now, in a normal scenario, in the meiosis 1, the two homologs segregate to opposite poles, and the two sister chromatids segregate to opposite poles in meiosis 2 normal situation. When non-disjunction or missegregation occurs, what happens is, for example, in case of true non-disjunction, the two homologs which are paired together are segregated towards the same pole, which leads to aneuploidy. Similarly, in achiasmate non-disjunction, which happens during meiosis 1, the homologs which are not actually paired together segregate towards the same pole, which again lead to aneuploidy. There can also be a case of premature separation of chromatids during meiosis 1, which could lead to polyploidy in one daughter cell and aneuploidy in one cell. Both of these cases of aneuploidy and polyploidy are deleterious to the organism. In case of meiosis 2, if non-disjunction occurs, which means that the two sister chromatids did not separate from each other as should be, and both of them are segregated towards the same pole, leading to aneuploidy. Aneuploidy and polyploidy result in either miscarriages or either result in genetic disorders. Coming back to the primary question, which is, if the siblings have the same parents, why are they different from each other? And how does this exchange of genetic material occur between the chromosomes? The answer to both of these questions lie in a process called homologous recombination, and we will be elaborating on the mechanistic process of homologous recombination in the next segment.